Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Meng, a professor of physiology and the director of the Center of Biomedical Research Excellence for the study of pain and sensory function at the University of New England. Ian's background is like many people I've talked to for this podcast. It took him a little bit of time to find his passion for science, but once he did, he was hooked. I learned a lot from Ian during our conversation and have a newfound appreciation for the eye and all it does for us. I hope you'll find some inspiration and new knowledge too. Today's episode is brought to you by Epstein Properties. Epstein was an axolotl sponsor of the recent Maine Discovery Museum Gala, our 90s party, and the whole Discovery team is grateful for their support. Ian, welcome to the Maine Science Podcast. It is a delight to have you. We first met, I want to say, in 2014, 2015, when I got to learn about all the great stuff happening at UNE. And shortly thereafter, you were part of a crew. You brought in a bunch of students and did a whole 2-day session on exploring the brain. I don't know if you remember it, but I remember it being A, super popular, and B, you all were exhausted when you did it.
Yeah, I mean, it was really exciting for us. And as you said, pretty exhausting as well. But up until COVID hit, we were still running an annual brain fair here at UNE on our campus, and where we open up the whole gymnasium to the community. And we have tons of booths, mostly, run by students, and they have a lot of hands-on activities. So we're hoping to get that going again soon, either this spring or in the fall.
That's really good to hear because it was really amazing. I think there's a lot that comes out of having students do that, not just for the students, but for the public. It's just great all around. But before we dive more into UNE, I thought I would give you a chance, like I do with everybody I talk to let us know how you got into science in the first place.
Sure. And I have to say that when I was in high school, biology was not my favorite subject at all. It was probably one of my least favorite subjects. It kind of came to me a little bit late in the game. So when I started college, I actually was more into music. I played the cello and was in the music program, orchestra, chamber music, all of that. I was really into political science, political philosophy, that sort of thing, my whole first year of college. And things really took a change, I would say, my sophomore year in the fall semester. They had a course that was an introductory neuroscience course that was open to people of all backgrounds. So you didn't have to have any kind of science background to take it. So for me at that time, it was really the perfect way to get into it. And because it was so accessible, it really brought me in and opened my eyes to how amazing neuroscience is and how much there's still to learn. For me, it was also really beautiful because I still remember, one of the first lectures during that time where they were showing some drawings that were made by Ramon y Cajal back in the late 1800s, where he was using this Golgi stain that would fill up these neurons. And then he would basically, using an ink pen and looking through an old microscope with just, one eye, essentially make these drawings of these neurons that were so detailed and so exquisite. To me, it was like art. And so that's really what brought me into it. And then from that point, it was really just finding a really inspiring professor who had a lab and had some openings in his lab. And then I joined. And, again, it was just sort of having a welcoming community that kind of brought me in, because I always felt like a little bit on the outside, not coming from a big science background and not really even liking it very much in high school. So it's been a pretty long road, but definitely worth it.
So you and I are about the same age. I'm curious how that introductory class would be different now, just because it feels like what we know about neuroscience has just exploded in the last 25 to 30 years. And I wonder if, maybe you can't answer this, but I wonder if, was it basic enough that it would feel similar, or would it be even more awe-inspiring, do you think?
Yeah, that's a great question. I've thought about that a lot because I teach in the medical school, but about 10 years ago or so, maybe closer to 15 now, I did start an undergraduate neuroscience program here, and I had to put together an introductory neuroscience course. And I tried to make it in a similar fashion, kind of something that was really accessible to all students. And I have found that it's really difficult to do, just as you said, because the amount of information has exploded. And I have to say that the reviews, as a matter of fact, I was just looking back at some of those reviews recently, The reviews from the students from those first few years was like, there's too much information for a one semester class. This needs to be broken up into 2 semesters. And in fact, that's what we ended up doing. It was just because I was essentially trying to cram everything into 1 semester from what I had learned maybe 30 years ago now, and it just wasn't possible.
I think it's a really good reminder to people that the student evaluations that y'all get actually mean something. So you take it into account. And I think it's really interesting because essentially the students there are saying you need to give us both less work and more work at the same time.
Yeah, that's right. I find that the students, they like to be challenged. They want to know that they're really learning something. At the same time, though, they don't just want a bunch of facts. And you can't just try to cram everything into their brain that's going to be gone in a week anyway. So I do think that diving deeper with sort of a narrower topics, fewer topics, but drilling down a bit deeper is definitely a way to go, even in an introductory class. I think there are ways to do that.
So you don't just teach at UNE, you also do research. And I know that you just got a pretty significant grant with the COBRA program, if I have that right. So I was hoping, number one, you could tell us what kind of research you do with neuroscience, and then tell us a little more specifics about your actual research now and that program too.
Sure. So why don't I start with the program and then I'll move on to my own
Fantastic.
research. Yeah, so the program actually is now in, I would say its 11th year. It's a program from the National Institutes of Health where they are funding programs within states that are traditionally underfunded by the NIH. And so in this program, there are a lot of acronyms here, and I'm not going to get into all of it, but it's called the IDEA program. And this particular grant that we have runs a center, and it's a center that focuses on the study of pain. And so that helped us to build research infrastructure. So it allows us to purchase a lot of cutting edge instrumentation and equipment that's needed to do the kinds of studies that we need to do. But it also helped us recruit a lot of faculty and to get their labs up and running and get them going on their own research programs. And that involves a lot of mentorship too, because when you start a new faculty position, when you start a lab, actually, a lot of it is like running a business. And you don't really learn that when you're in graduate school to be a scientist. And so, you're managing people and you're managing budgets, and you have to figure out how to, keep your science going at the same time. So you're certainly juggling a lot of different things. And so part of the center is to really help provide that kind of support to teach people those things that they don't learn in graduate school. And I had to learn sort of the hard way, kind of going through the process, for the first, second, and third time. So that's what the center is all about. And I have to say, it's been really transformational at the institution here at the University of New England. We've hired a number of faculty as part of this center. We really have a great cohesive group of investigators now. We do a lot of collaborating, which is the way science works now. You have to be collaborating with people to really move things forward. And so I think that, the sort of the time and effort put into that center has really been well worth it for me. In terms of my own research, that work, if I were to go back to that first lab I was in as an undergraduate student, that lab actually worked with stress. And it was all about different models of stress and how stress might actually be something that leads to depression. Because in the model that they were into at that time was called learned helplessness, where essentially you have stress that's uncontrollable, that you have no control over, and all of the different kinds of effects that would have on the brain and behavior. And so that was my first lab. And when I was looking at where to go from there, I always saw pain and chronic pain, especially, as a big source of uncontrollable stress. And so that was sort of my avenue into that world of pain and chronic pain, especially. When you enter the world of pain, it's so big. And in a way, it's kind of like, what's your niche going to be? You have to sort of find a home for yourself. And I started out really working with pain that has to do with the head and face. And there you're dealing with a lot of things like migraine headache, tension headache. So a lot of headache, but also temporomandibular joint pain. So TMJ pain. There are dental pain. So a lot of pain investigators who do what we call trigeminal pain, because these are areas that are innervated by the trigeminal nerve. And that's the main sensory nerve that innervates the head and face. So a lot of the investigators in that area, in that field, are actually dentists or in dental schools, as you can imagine. And so some of my research in the past has been related to headache. But as a graduate student, I actually started focusing on work that was being performed by my mentor in graduate school, which was dealing with corneal pain. And when a lot of people think about that, they're like, that's something I never even thought about, is pain from the cornea. And the cornea is the outermost part of your eye. And I have to say, most people think of pain as being a bad thing, but pain is really there to help. It's there to say, hey, there's something wrong here. You need to protect this, or you need to get away from whatever's producing that pain. And so for the cornea, as being on that surface with the environment and there to really protect your eye, the pain signals are so important, essentially, to keep you healthy and to keep you able to see. So if you are unable to feel pain on the cornea, then you're going to injure your eye and you'll become blind. And so the cornea is actually the most densely innervated structure in your body. And all of these nerve endings come right out in between the epithelial cells, the outermost layer of the cornea, and they really are there to sense the environment. And part of it is to certainly sense anything that might be dangerous. So whether it's an eyelash or whether it's rubbing your eye after chopping hot peppers, cutting an onion, but at the same time, I would say that there are a lot of things that those nerves are doing that are unrelated to pain. And I think that's something that has been less appreciated. But if you think about the surface of your eye, you have tears. You're always blinking, of course, to keep the tear film coating the surface of the cornea. But the neurons have to be able to sense when things become dry and when you need more tears and when you need to blink. And so you have all these what we call more homeostatic functions of these neurons as well. And so part of the puzzle really has been, how do you tease apart the neurons that are involved in those more homeostatic functions from the neurons that are giving you that pain signal? And so that's kind of what we've been focusing on in the lab, because there are a lot of people out there who have dry eye and dry eye disease. So they have chronic dry eye where it feels like when you blink that there might be sandpaper on the back of your eyelid. So that lubrication is really important for you to not feel any pain and for the health of your eye in general. And so you have essentially this process where these neurons, when the cornea becomes dry, these neurons become more active to produce more tears, but also the neurons that are sensing that, things might not be right here, that pain signal, those neurons are also becoming active. And so when you give a treatment for dry eye or for chronic eye pain, you want to essentially be able to shut down those neurons that are involved in producing that pain signal, but you don't want to shut down the neurons that are involved in all of those other functions, because you want to keep producing tears. You want to actually drive tear production, because that could be helpful, but you don't want to have all those pain signals coming in as well.
How do you do this explicitly? I'm fascinated at how you could make this happen.
Yeah, I have to say that the tools we have now are just increasing exponentially over time. Most of the work involves rodents in our research, and so it's mostly animal research. And certainly, we're very sensitive to the fact that we might be producing some sort of pain in an animal. And so we always like to use stimulation that might be very brief in nature. So hypertonic saline, sort of putting saline in your eye that might have too much salt in it. And then you essentially can see a reaction to that, which might be tearing or blinking or wiping at the eye for a very brief time. So we need to have some kind of behavioral output to tell us what the conditions are like. And so that certainly is a component of how we do our work. The main thing is we want to relate neurons and the activity of neurons to some sort of behavior. And whether it's a pain-related behavior, like a wiping of the eye or blinking or squinting of the eye, those are things we can measure. We can also measure tear output. So we could put like a little cotton filament in the corner of the eye for a period of time and then measure the amount of tears that are being produced that way. So we can then take those sorts of behaviors and then correlate them to what's going on with the activity of neurons. And for that part of it, there are different ways to get a window into the brain, you might say. So we can actually put electrodes into the brain, insert electrodes into different regions and measure the electrical activity. And we call that those action potentials. And so with those electrodes, we can then look at the properties of those neurons and how they might change with different conditions like dry eye or with these different kinds of stimuli. There are also ways we can do some imaging as well. So we can take tissue from the brain or the cornea itself, and we can label them or stain them for different proteins, or we can look for expression of different genes and how the expression of those genes might change with different conditions, and then try to see how that might affect the activity of neurons, and then see how the neurons would affect the behavior. So we kind of try to go, sort of that whole spectrum from gene expression all the way to some kind of behavior. We actually used to do most of our work in rats. I have to say, now we've really moved to the mouse models. And the reason for that is because of the transgenic mice that are available. And those are mice where you've been able to go in and change the genes in such a way that you can have specific neurons expressing different kinds of, a lot of times we use fluorescent proteins. So a green fluorescent protein, which was taken from a jellyfish, and now it's inserted into neurons in a mouse. And then we can visualize those neurons. And so then we can look at morphological changes, how the shape of those neurons might change. But then there are things like calcium sensors. So proteins that will essentially tell you how much calcium might be inside of a neuron. And then we can essentially look at sort of a way of looking at activity, because when neurons become more active, then more calcium comes into the cell. And so those are some of the other ways that we can kind of get a window into what's happening.
And I'm betting that the mouse models, they translate in some way to human models as well, so that what you're learning there, you can at least feel some confidence that the same thing is happening with humans.
Yeah, absolutely. And that's always a big question. So, I have to say that one of the projects that I'm working on right now is we're trying to look at the whole diversity of neurons that are innervating the cornea. So, all of these different subtypes of neurons and all of the genes that they express for individual neurons that are innervating the cornea. And can we kind of clump them into different groups based on gene expression profiles? And we're doing that in the mouse. But then I have a collaborator who's also getting some human tissue from the trigeminal ganglia, which is, again, the trigeminal nerve coming back. So it's the cell bodies from those nerves that are innervating the cornea, to see whether in the humans we can find those same populations of neurons, so we can kind of relate one to the other. But you're right, that's always something we're thinking about, is how does this relate to the human? And I have to say, with respect to the cornea, obviously there's a clinical problem that we're dealing with here, right? I mentioned dry eye. There are also people who might have corrective eye surgery, so LASIK surgery. I notice the FDA now is saying, hey, maybe there should be a warning about this kind of surgery, because sometimes it does produce chronic pain afterwards, or chronic dry eye after that surgery, because essentially what you're doing is you're cutting all of those nerves that are innervating the cornea. They do grow back, they reinnervate typically, but you don't know that it's going to have the same function after it reinnervates the cornea.
Have you been able to start to make the distinction between what you were saying, the parts of the cornea nerves that identify pain and then the other ones? Like, have you been able to map those with the work you're doing?
Yeah. So here's what we think right now, anyway. When we were first looking at this, I thought, oh, this is a real breakthrough. I think that especially when you're looking at the cornea, the ability to translate what you get in a mouse model to humans is really great, because you're essentially dealing with a structure that is right out there on the surface. There are a lot of topical treatments that you can give for corneal pain. And of course, you don't want to just give an anesthetic, because that's going to basically reduce the activity of all of those neurons. And so you have the potential for self-injury. So how can we identify neurons that are just involved in pain and those that aren't? We think that we know something about that, in that the neurons that look like they're not involved in transmitting the pain information are exquisitely sensitive to drying of the corneal surface, of the surface of the eye. So we noticed this sort of by accident. We had an electrode down in the trigeminal ganglia, recording from one of those neurons that's innervating the cornea. And then we happened to be doing something else in the lab at the time. And then we noticed the neurons started becoming very active. And it became very active because we essentially forgot to put some artificial tears on the eye, and it started to dry out. And so then the neuron started to activate from that. And what we realized soon afterwards was that those neurons were very sensitive to a mild cooling of the corneal surface. And so if you think about it, it makes complete sense. But we hadn't thought about it at the time. Essentially, when you have tears on the surface of your eyes, just like when you sweat, when you have evaporation of those tears, you get evaporative cooling. And so that's the signal essentially for saying your cornea is drying out and you need to produce more tears. So it's ongoing in everybody all the time, but we're just not consciously aware of that happening.
I love the fact that it was by accident. You were doing something else. I think people I've talked to, that happens quite a bit. And so the key there is to pay attention to whatever the messages are sending. It's fascinating that it's something that's so standard with the evaporative cooling that you don't think about till you see it right in front of you. And then you say, oh, of course. That's really interesting. It's almost this alignment of you getting to go like, oh, why wouldn't it work like that?
Yeah, that's right. And so what that led us to was a protein that's expressed in those neurons, which is called TRPM8. And so this is part of a family of proteins called TRP channels. And so it's TRP channels. And so a lot of people are familiar with TRPV1. TRPV1 is actually the receptor for capsaicin. Capsaicin is that ingredient in hot chili peppers that make it feel really hot. So neurons that sense heat or noxious heat, those neurons are expressing TRPV1. And that's why when you eat something that's really spicy, it feels hot. So neurons that are sensing things that are cooling or mildly cooling, those neurons are expressing TRPM8, which is sensitive to menthol. So that's, again, sort of a compound that's produced by the mint plant, and it feels cool when, you're chewing your gum or something like that. So neurons actually expressing this TRPM8 channel, they're very sensitive to this cooling stimulation, which is essentially because of the evaporation of tears. And we thought, okay, that's great. Now we have a target so we can increase tearing without producing pain. I thought this is great and this could be a breakthrough. And I was so naive at the time. So the reason why is because next we essentially took a look at what happens when you have dry eye and what sorts of changes or alterations might there be in neurons when you have that condition of dry eye. And it turns out that it looks like those neurons that express TRPM8, when the eye becomes dry chronically, their expression patterns change. And so not only do they express TRPM8, but they start to express V1 as well. So you essentially get neurons that are now sensitive not just to the mild cooling, but also appear to be sensitive to capsaicin and things that are noxious in nature. And so we think that maybe the function of these neurons might also be changing too. So they might start to transmit information that may be perceived as painful. So that's something that is ongoing, and we're not exactly sure about that, but we have some pretty good indications that that may be the case. The other aspect to it, which also makes it more complicated, is that those neurons also become much more sensitive to things like menthol and to the cooling. By more sensitive to it, I mean they initially become much more active when you apply menthol to the cornea. But because they're so sensitive to the menthol, they then desensitize. And so what that means is they become less sensitive because of the exposure. So they actually shut down because they're so sensitive to it. And that obviously has some implications if you're trying to figure out a nice treatment. Activating a neuron that's sensitive to something like menthol. Now suddenly you apply the same concentration of menthol, and instead of activating the cell, it's actually giving you maybe a short burst of activity, but then it's becoming completely silent. So that was something that was really unexpected for us. But certainly, and that's science, just like there are always things cropping up like that.
Do you know if that happens with other neurons in general, the oversensitivity and then just shutting down, or is it specific to what you're looking at?
It's funny. I think that it does happen to other neurons. And I would say that, and this is something that's kind of interesting in that if you look at all of the sensory systems, whether it's the visual system, auditory system, taste, touch, if you walk into a loud room, you acclimate to that. If you taste something and then you taste the same thing again and again, then it becomes less intense each time, right? When you put on your shirt, you might feel it at first, but suddenly you're totally unaware that you're wearing a shirt. Otherwise, it'd be totally annoying to be wearing clothes, right? So I think that that's something that's actually common in most of the sensory systems. Pain is actually the exception usually. So with pain, typically, if you injure yourself and then you poke that place that's been injured, the pain ramps up and it gets more intense each time you stimulate. And so a lot of the focus of pain research is sort of what causes the signal to keep increasing rather than adapting like most of the other sensory systems. As a general principle, I think that that's often the case, that things will adapt. But even in the pain system, if you give a very high concentration of capsaicin, that TRPV1 compound that activates those pain fibers, if you give a very high concentration, then again, you might get that burst of activity, but it also desensitizes those pain fibers. There actually is a clinical application for this. So if you actually go to the pharmacy, you can buy creams that have capsaicin in them. And oftentimes it could even be prescribed for something like herpes zoster. So something where it's a very painful condition and you want to apply something topically to desensitize those neurons that are innervating the skin. And so you can give, especially when you give repeated applications of a very high concentration of capsaicin, you might feel initially a brief burning sensation, but then you can get the desensitization.
And does that stay, the desensitization, or is it limited?
It can last for quite some time. When it comes to capsaicin and the high concentrations, a lot of it is concentration dependent. How high is the concentration? But if you give a high enough concentration of it, then not only do the neurons desensitize or become less sensitive, the nerve endings will kind of die back a little bit. And so for that reason, it can actually be a pretty long-term kind of effect.
I don't want to let you go without giving you the chance to explain how students work in your lab. I know you're not doing this all by yourself, that you have students who help you out. And I know it's collaboration as well. But I think one of the greatest things personally about having undergraduate and graduate students is it gives them a chance to learn how to do science, and certainly as undergrads to learn if that's something that they want to do. One of the best experiences I had as an undergrad was to work in a lab because it convinced me 100% that that is not what I wanted to do. And that is super valuable to learn. So I was hoping you could possibly explain just a little bit how what you're looking at gets broken down so that students can help both advance the science, but also learn how to be a scientist.
Yeah, absolutely. So first, I love having undergraduate students in the lab. I have 4 currently in the lab, and COVID was hard for that reason. It was really hard to have students in the lab at that time. And so coming out of COVID, we've been ramping up, and that has been difficult because when I have students come into the lab, I don't just, have them go off and do their thing. There's a lot of intense training that goes with that. And so some of that's done by me, certainly, but also I like to pair students up, sort of a more senior student with a student who's just coming in and have them work together on a project. I find that it's always helpful, but it was hard when everybody was new because there was nobody in the lab during the COVID period. But I think we're getting on it. We're doing well right now. Last semester, there's a lot of training going on. So students, I don't expect them to have a lot of background. I don't expect them to worked in a lab before or know much about research. The whole idea of it, as you said, is to kind of get that exposure and know, hey, is this for me? So when I always talk to students before they start in a lab, just in the conversation, it's all about what's the best match for them. Every lab kind of has their own personality, is kind of how I look at it. And I'm always encouraging students to see everything that's available. Not only the kind of research they're doing, but talk to other students in the lab. What is it like? And what's the environment like there? And even if you start to work in a lab, it doesn't mean that that's the only lab you can ever join. If it doesn't work out, if you feel like that's not the thing for you, that's fine. And certainly for me, I don't take it personally. And I don't think any lab director should take that personally. It really is finding the best match. But once in a lab, I think that, again, it's sort of learning by doing. And they always say sort of show one, do one, teach one. You just have to be ready to jump in and try it after watching it, maybe even just one time. And then we also have regular lab meetings. So we'll present papers and talk about the projects. And again, for people who are just starting the lab, they may be completely lost when we start. And I let them know, hey, no question is a dumb question. It's okay. And if you just sort of stick with it for a little bit, then you'll start to catch on. And then by the end, when they're a junior or senior, they'll be presenting the papers themselves and kind of talking about the research and what it means, and have maybe their own project that they're working on and trying to complete before they graduate. So it really is an evolution, kind of taking somebody from the very first stage all the way through to some independence.
So I think one random question that just came to me, with you focusing so much on pain and eye pain in particular, has it affected how you think about your eyes yourself? Like, do you sometimes go like, oh no, what am I doing to my cornea? Or has it made you realize that you don't have to worry about it so much?
Yeah, I mean, certainly, I think that it does make me appreciate the importance of the cornea in protecting your eye. And that's something that I think it really has demonstrated to me. But I would say that I probably won't have LASIK surgery to correct my eyes. Just, me personally. The funny thing is that the eye has always made me a little bit squeamish. And I don't know how you feel about that, but and I know there are other people too, where when you see something happening to the eye, even if you're looking at an eye surgery going on or something like that, you just want to close your eyes and rub your eyes a little bit. That feeling hasn't gone away for me.
I find that oddly helpful. Out of all the critical sense organs, I think the one I am most focused on is the eye because it really was super limited, right? We've only got the two. And why would you mess up the, taste? Sure. I think you're more used to taste coming and going, not to dismiss people who have issues with that, but it seems more repairable in some ways than the eye. So I'm kind of with you. I was wondering if you'd gotten just your own desensitization to it, or it's just made you even more aware of how remarkable it is.
I think probably more aware of how remarkable, for sure. Yeah.
Well, Ian, this has been great. I really appreciate your time. I never get tired of the people who I get to talk to about their work, and you all are so excited and happy talking about it. You have been smiling for most of this conversation. And I think it's all the more remarkable when I think about the fact that you're dealing with research about pain. My first thought is like, oh my gosh, how hard is that to deal with every day? But recognizing that what you're trying to do is break it down so that people have less of it, I totally get why you're smiling most of the time. So I really appreciate both your time here and the work that you're doing.
Well, thanks, Kate. It's been really great to talk to you.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is hosted by me, Kate Dickerson. This episode was edited and produced by Scott Loiselle. We received financial support from Central Maine Power, production support from Miranda Bouchard, and social media support from Next Media. The Discover Maine theme was composed and performed by Nick Parker.